Hey Happy Bio Bruins, and we're going to talk about proteins. And here's a little animation, and here you have a model of a protein. Now proteins can be very complex, but we can simplify it by analyzing the different parts of it. Now proteins are made of many different types of chains. So here, for example, we have myoglobin, and myoglobin carries oxygen in our muscle cells. And what allows myoglobin to carry oxygen? It's the structure. So here's another protein that turns on and off genes, and as you can see, this protein has a very different shape from myoglobin, and its shape allows it to grab onto the DNA. So again, proteins play many roles in our bodies. Makes part of our structure. They are enzymes. They're part of our immune system, antibodies. Some proteins are hormones. Here's another example. This protein is called porin, and notice it looks like a protein. It's like a channel, and again functions like a channel due to its structure and shape. And if you look at the molecular chains, it's composed of chains, and if you see there, you can see where it starts, and it's got a lot of kinks, and each kink represents a chain. And if you look at it closely, it's like a chain that's like a bracelet, and these chains are determined by the side chains, and each side chains, and basically the building block of a protein are amino acids. And then here's porin again as a whole protein. So when we study proteins, we look at what they're composed of and what are the different parts. So proteins again vary in structure. There are diverse roles in our body and every living organism. Here you have an enzyme. Anything that ends with ASE is an enzyme. Some hormones are proteins. Our structural, like muscles, skin, are also made out of protein. Your hair, your nails, and here's another important hormone, insulin. Now, the building block of a protein is called amino acid. So an amino acid. So here's an example of an amino acid, and it's what we call the monomer. Now, look at the different functional groups an amino acid has. It has an amino group, that nitrogen and 2 hydrogens, and it has a carboxyl group, and what we call a side chain. And this side chain, there are 20 different kinds, so there's 20 different amino acids. Gives the property of that particular amino acid. Now, when you want to combine 2 monomers of protein or 2 amino acids, remember to form bonds, we form it by dehydration. So when we remove a water molecule, that forms a bond between a carboxyl group of 1 amino acid and a amino group of another amino acid. So when they form a bond, this bond is called a peptide bond. So here you have 3 amino acid, or we call it peptide chain. Now, like I said, there's 20 different amino acids. You don't need to memorize this, but these amino acids have different properties based on their side chains. So if you see this shaded in white, that's their side chain. So each amino acid has an amino group, a hydrogen, and a carboxyl group, and a side chain. So these side chains can be nonpolar, that means it has no charge. Their side chains can be polar, and notice a functional group, the oxygen and hydrogen, makes it apolar. And here you have sulfur, a sulfur group, and amino group. And in some amino acids are charged. They can be acidic or basic. Acidic meaning they accept they donate hydrogen, and basic accepts as a hydrogen acceptor. Now, proteins have different levels of structure. The first level of structure is called the primary structure, and basically it's a sequence of amino acid, or we call a peptide chain. So think of it like a pearl necklace where each bead is made up of different amino acids. Now, if you take that chain and twist it or pleat it, you'll have a secondary structure. So there are basically two different types of secondary structures. One is called helical, and notice it becomes a helical structure because you have hydrogen bonding between the amino group and the carboxyl group, and it does so in a helical fashion, so we call that a alpha helix. Or it could be pleated, so we call it a beta pleated, and again you're forming hydrogen bonds between the amino group and the carboxyl group, and that's what we call secondary structure. Now when you have different secondary structures combined, and you have what's called a tertiary structure, and this tertiary structure will also have disulfide bridges. So for example, in your hair, someone who has curly hair, it's curled because of these disulfide bridges, and usually disulfide bridge is an example in tertiary structures. So you can have also other kind of interactions going on, like hydrophobic interactions or van der Waals interactions. They also contribute to the tertiary structure. Now, quaternary structure is a combination of secondary and tertiary structures put together. So here, for example, you have collagen, okay, and you have hemoglobin. So notice you have different tertiary structures combining together to form the quaternary structure. So protein structure is again determined by the sequence of the amino acids. So the different types of amino acid is like the recipe for whatever protein you have, and its shape. And its shape is again determined the interactions between the R groups, forming that 3D conformation. So we usually, when you have a primary structure, that protein is not functional until it has this 3D shape, then it becomes functional. So if you have R chains that are hydrophobic, usually you'll find them in the center of the protein. And if you have side chains that are hydrophilic or polar, usually you'll find those on outside of the protein. Now, protein structures are very sensitive to temperature and pH, and they can be altered if the pH is not right or the temperature is too warm, and that's what we call denature. Now, some proteins, once they're denatured, you cannot reverse it, and some are reversible. So for example, when you make egg, once you cook it, you cannot reverse its structure. So if you alter a protein structure even by one amino acid, it'll totally change its shape. An example of that is sickle cell anemia. So sickle cell, in normal red blood cells, you have a nice donut-shaped cell. Now what happens is, if one amino acid at position 6, you have glutamic acid, becomes valine, what happens is that cell sickles. The shape totally changes, and then therefore cannot carry oxygen. So even one change in that amino acid can cause a whole different shape. What determines amino acid sequence is determined by your DNA. So the DNA is basically the recipe for proteins. So here you have a DNA strand. If you remember protein synthesis from DNA, that's transcribed to mRNA, and that recipe in the mRNA goes out to the ribosome and puts together the sequence of amino acids. So another important proteins are hormones, and these are some modified amino acids. So here, for example, you have tyrosine. When you remove the carboxyl group and additional two hydroxyl groups, you have another messenger called norepinephrine. And if you add a methyl group, it becomes an epinephrine. Okay. Here, for example, you have thyroxine, which is found in your thyroid, and this enzyme is in cells. And then if you add, if you remove another iodine group, then it becomes T3, which is more active. So take this from the PowerPoint and summarize what you know about proteins now. So proteins, what are they composed of in terms of elements? Proteins have carbon, oxygen, hydrogen, and nitrogen. A monomer protein is called amino acids. Can you draw an amino acid? There's a central carbon group, has an amino group, has a R side chain, hydrogen, and a carboxyl group. What's the polymer of a protein called? Polypeptide. Also, a functional polypeptide is called a protein. And what kind of bonds do you find between two amino groups? And yes, it's called a peptide bond. So properties, structures, function. So proteins are important to us because they're part of our structure. For example, muscle, they're part of skin, hair, nails, and so forth. They are part of our amino acid, immune system, like antibodies. They're messengers, like hormones. Most enzymes are proteins, so they conduct most of the chemical reactions. And what else? So proteins play a major role in our body. And the structure of a protein again depends on how that amino acid chains are folded. And one thing you need to know is the different types of interactions. So for example, you have hydrogen bonding, you have disulfide bonds, you have van der Waals interactions, you have hydrophobic interactions. So make a whole chart on the different types of interactions, and what type of interaction depend what type of structures. So for example, what kind of forces do you see in a primary structure? So in a primary structure, for example, the sequence of amino acids play a major role. In secondary structure, what plays a major role, and that's usually hydrogen bonding. And in tertiary, you have disulfide bonds, also hydrogen bonding, van der Waals interactions, okay, and so forth.